Welcome to the course on medical biomaterials. Today we are going to talk about animal studies, that means in vivo studies, and animal studies are supposed to be carried out before they actually test it out on human volunteers. So, you want to look at the biocompatibility of the material under in vivo situations. That means we want to look at biocompatibility, safety in biological environment. Whatever we do in the cell culture lab in vitro, nothing like performing it in animals, because animals are the real system where we have a lot of enzymes, we have the blood, we have the other body fluids taking part. So, the way the biomaterial behaves in such environment may be completely different the way it behaves in very controlled in vitro. There are guidelines for in vivo assessment of the tissue compatibility given by FDA, regulatory bodies, ASTM, ISO, USP. So, there is ISO standard 10993. It provides guidelines on biological evaluation of medical devices. Part 1 gives the guidance on selection of tests, part 2 gives the animal welfare requirements, part 3 gives the guidelines for specific test procedures. Okay. Now, if you look at the Declaration of Helsinki in 1964, okay, it mandates that medical research on humans must be supported by preceding animal research. So, before it goes into human volunteers, there has to be experiments conducted on animals. Okay. So, this has been taken very seriously and they made it into a law in USA by the FDA and Europe by the European Agency for the Evaluation of Medical Products. So, animal trials is a must before it goes into the human volunteers. So, what are these in vivo tests for biocompatibility? There is something called sensitization test and irritation test. That means, does the biomaterial cause some irritation to the animals? Does it become very sensitive to certain body parts or tissues? It is not only the material, but also the extracts from the material. Okay. So, things like skin test, mucous membrane test, intracutaneous test. So, there are different types of tests, basically looking at whether it causes irritation and sensitization to the animals. So, that means that does the material contain leachable chemicals that may cause adverse local or systemic allergy or hypersensitivity reactions on exposure for prolonged period. So, that is what is called sensitization test and irritation test. Okay. Then comes systemic toxicity. Does the material gives overall toxicity to the animal, like acute toxicity, subacute toxicity, subchronic toxic, not only the material but also the leachable. So, you need to understand it is not only the material, but whatever is getting leached out from the material also has to be studied in detail for all these. Generally mice, rats or rabbits are used for this systemic toxicity test actually. The animals are observed for any toxic signs immediately after injection and for a set time point for so many hours, short duration, long duration, and then one studies whether it causes any toxicity to the system. Then subchronic toxicity, this is generally used to determine the toxic effects on long term application. Okay. One is short, other is long term, especially we need it for permanent devices like devices which like stents for example, or orthopedic implants or knee joints, diaphragm valves, okay, heart pacemakers. All these are going to be permanent devices. So, the materials involved, are they going to have any lead to any subchronic toxicity? Okay. So, short term toxicity, long term systemic toxicity. So, the various in vivo tests for biocompatibility include sensitization test, irritation test, acute systemic toxicity test, subchronic toxicity, genotoxicity. Okay. Does it cause any problems for the genes? Implantation test, does the material still perform when it is inside the body? Hemocompatibility, does the material cause any problems to the blood or is it compatible with the blood? Carcinogenesis, does it produce any cancer or carcinogenic effect to the animal? Does it going to cause any reproductive toxicity or developmental toxicity. These are much longer. They look at the next generation of animals and one need to look at it. So, pharmacokinetics, this is very valid, especially when you are talking about drug delivery system, how fast the material goes from the, for example, the oral into the bloodstream and how fast it gets eliminated. So, mostly it is for drug delivery systems, but not for permanent implantable systems. Or if there is a biodegradable polymer, how fast it gets degraded and disappears from the body. Okay. Preclinical safety testing. So, all these are histopathology. Then later on the various tissues from the animal is taken out. It could be related to the kidney, liver, various body parts like heart, and then when the doctor, the pathologist looks at changes and the doctor also looks at whether there are any inflammation that is caused because of this material. So, these are a large number of tests, and you need to remember that it is not that you need to perform all the tests for all the biomaterials. So, some biomaterials may require some tests, some other biomaterials may require some other tests. Okay. So, what is this genotoxicity? So, you need to identify, does the material lead is a potential genotoxic effect, that is changes in the DNA, does it mutates some genes, so that the substance directly or indirectly induces genetic damage, that is what is called genotoxicity. That means the material changes the genetic makeup either directly or indirectly. So, this may involve in vitro, I can do it in cell lines and I can do it on animals also. Okay. Then comes Ames test. It detects point mutations by employing this particular bacteria called Salmonella typhimurium. Okay. So, if there are any mutations in the gene at which place it got mutated, and because of that what could be the long term effects, that is called the Ames test. Then look at the lymphs, changes in the lymph pattern, these are all you can do it using mammalian cells to detect the point mutations. Okay, that these are related to gene. It also includes chromosomal aberration and changes in the mouse micronucleus assays. So, these are animal trials as well as some of them could be cell line based work. So, all these lead to understanding whether the material causes genotoxicity. Okay. Then comes the implantation test, material is placed inside the body in different places, and one needs to look at whether it is biocompatible when it is directly in contact with tissue other than the skin part of it. So, if you are talking about sutures, implantable material, one needs to understand that. So, you need to also test whether the material performs its job both in the short term as well as in the long term. Okay. So, various implantation techniques, various locations inside the body in which the material is kept and it is studied. Okay. This provides information on the safety of the material. Then looking at carcinogenesis, you are determining the carcinogenic potential of the biomaterial. That means whether it will cause cancer, whether the leachables cause cancer over a period of time. So, if the biomaterial is going to be permanently kept, so maybe you may have to keep the material for a long period in the animal and then see whether it is going to cause carcinogenesis. So, it could be carried out over the lifespan of the material. It is expensive and it could be controversial also. We are not very sure whether what the results coming out of the animal studies can be extended to human, as well as lifespan means do you really run it for very long period of time and so on actually. So, it could be controversial. Now, there are a lot of tests are done as per the guidelines based on the intended use of the material. Okay. So, we have surface devices, that means your skin patches or wound healing patches, external communicating devices. Okay. So, like your pacemaker, these are all externally communicating, biosensors, implanted devices like your cardiovascular stent, urethral stent. So, surface devices means it could be in skin, it could be in mucous membrane, or it could be just breached or compromised surface. That means you make a very surface patch slightly deeper. Okay. If you look at external communicating device like blood path, indirect tissue, bone, dentin, circulating blood. Implantable could be tissue, bone, blood, all these. Okay. So, the duration could be less than 24 hours, 24 hours to 30 days, greater than 30 days. So, generally if you look at papers and look at what type of studies people have done, you will see either they would have done for 24 hours or they would have done for 30 days. So, limited is less than 24 hours. So, if you want prolonged, you may run it for 30 days in the animal and then look at the effects, or if you are looking at permanent devices, you need to run at least for greater than 30 days. Okay. So, you have different situations here, ABC, ABC, ABC and so on, and then you are looking at cytocompatibility. Is it causing sensitization? Does it cause irritation? Especially if it is a very small duration or even if it is long duration in the skin, we are satisfied with these three different tests on animals. Okay. Please note, this table was collected from Biomaterial and Biocompatibility Testing Lab, WHO, India. If you are looking at slightly mucus, then we not only do the irritation and sensitization, we also have to look at subchronic toxicity and genotoxicity. Okay. Similarly, if it is slightly breached, we may again have to look at subchronic and genotoxicity. Now, if you are going to have external communicating device, that means the material is somewhere in the blood path, then we may have to look at hemocompatibility also. Please note this, okay, and systemic toxicity, even if it is a short duration or whether it is a long duration. If it is a long duration, we also have to look at chronic, genotoxicity and hemocompatibility. Okay. Look at this. So, the number of tests increases. So, if it is a skin, and tests are mostly irritation, sensitization. Okay. If it is slightly longer duration, we may have to look at chronic toxicity and genotoxicity. But if it is in the externally communicating device, it is in the blood path, we may have to do the hemocompatibility. If we have to look at tissues, then we have to do a lot of other tests, systemic toxicity, chronic toxicity, genotoxicity, then implantation test. Same if it is a blood related, then of course we do the hemocompatibility. Okay. So, if it is an implanted device in a tissue, again we can stick to only irritation, cytocompatibility and sensitization, but if it is down into the bone or blood, we may have to do the entire set of tests here. That means not only the sensitization, irritation, cytocompatibility, we have to look at systemic toxicity, we have to look at subacute, subchronic toxicity, genotoxicity, implantation test, hemocompatibility. Okay. So, this is a very interesting table, the type of test that need to be done depending upon whether the material has to be for limited duration, that means less than 24 hours, for prolonged duration, that means 24 hours to 30 days, or permanent greater than 30 days, and whether it is a surface device, whether it is an external communicating device, or whether it is an implanted device. So, the number of the type of test changes depending upon these two factors, and these are the various types of tests that needs to be done. Okay, these are the guidelines that has been suggested by WHO India here. Okay. Interesting table to look at. Selection of animals for in vivo studies. Okay. So, there are something called small animals, there are something called large animals. So, small animals could be rats, mice, rabbit, okay, hamster and so on. Large animals could be pigs, sheep, dogs, even horses. Large animals are very expensive to work on, small animals are much cheaper. In some situations we have to work on large animals because small animals have very small cartilages, for example. If you are looking at cardiovascular stents, the veins and the system is so small, we will not be able to do the stent studies in small animals, we may have to go to large animals. So, if you are looking at heart valves, we have to go to sheep, we cannot do it in small animals. Stents, again we have to go to pigs and dogs. Artificial hearts, we have to go to really large animals, calves. vascular grafts, dogs and pigs. Bone regeneration, rabbits, mouse, rats. So, we can do it in small animals, but joints we can't do in small animals. You see the joints of small animals are so small, we have to go to dog and goats. Cartilages, we have to go to rabbit and dog. Tendons, ligaments, again large, dog and sheep. Neurological, peripheral nerve regeneration, rats, cats. Electrical stimulation, rat and cat. Contact lenses, rabbit, because eyes are quite small to test. Intraocular lenses, rabbit and monkey. So, a large set of animals are used in preclinical studies, and depending upon whether we are looking at simple cytotoxicity, biodegradability, or if you are looking at the cardiovascular stents, heart transplants, or heart valve changes or joint changes, we may have to go from small to larger animals. Okay. Again, this is a very interesting table to get a feel of what type of animal studies that need to be done. Okay. So, the selection of animal models depends on so many factors. Okay. This is taken from this particular reference. Appropriateness as an analog. Is it appropriate to use this particular animal? Okay. That's the is it correct to use that animal, or should it be is the knowledge we get is too far away from the real human system? Transferability of information. Can I transfer this information I got with this animal study to what I want to achieve later? Genetic uniformity of the organism. So, if I am taking say Vista rats, genetically it will have uniform pattern, genetic makeup. Background knowledge of the biological properties. So, we know all about, for example, some of the animals we have shown, we may know all about Vista rats. So, immediately Vista rats are used for many drug discovery programs, because the biological properties of those are very well known. Okay. For example, if in bacteria E. coli, the biological properties of E. coli and the genetic factors are very well known. So, that is why in many studies you will see E. coli. Yeast, for example, is well known. So, many studies are done on yeast. Cost and availability. Cost plays a very important role. If you are talking about animal studies with sheep and dog and horse, it could be very expensive. Availability. Is it easily available? Because when I am going to do experiments, I may have to do it on many samples, so that it is statistically significant. So, the availability also important. Generalizability of the results. So, whatever results I get, can I generalize it for many other conditions? Okay. I might not be able to do for all conditions, can I generalize it? Okay. For example, I am looking at say a wound healing patch, I test it out on some animal, can I use the same knowledge, say for example, for burn wounds? Can I use the same knowledge if it is a diabetic wound, if it is a infected wound, and so on. So, that is called generalizability of the results. Ease and adaptability to experimental manipulation. So, I am going to do a lot of experiments on the animal. So, are the animal adaptable to the conditions? Is it easy for them to acclimatize? Ecological consideration. So, what are the ecological factors that come into? Ethical implications. What are the ethical implications involved? Okay. What is the ethics of using so many animals, and so on actually. So, all these need to be considered when we are selecting an animal for our preclinical study. Okay. For example, if I am doing cartilage repair, okay, cartilage you all know, these defects cause morbidity for patients, and it becomes very difficult for surgeons trying to repair cartilage. Sportsmen get this, old people get this, especially those who have a lot of physical activity get this. So, nowadays a lot of thought is being given on use of biomaterial for repairing this. Why it is so difficult? Because avascular nature of the chondral surface and the specialized rigid extracellular matrix with a low cell density. So, this tissue rarely regenerates by itself. Okay. This tissue does not, because it has got a low cell density, okay, it has got a specialized rigid extracellular matrix, avascular nature, it is not a vascular system. Okay. So, What are currently they do? Try to do bone marrow stimulation, cartilage plug transplant, and expanded autologous chondrocyte implantation. So, different types of that means they try to implant cartilage, they are trying to do bone marrow stimulation and so on, but with little success. So, when biomaterials are being looked at using artificial cartilages, you need to do animal studies. So, you need to plan based on the size of the joint, the cartilage thickness, the depth and critical size of the defect. A defect which will not heal spontaneously without any intervention. Okay. If there is a small defect, it may heal spontaneously, but if it is a large defect, it will not heal. The age of the skeletal maturity, that means generally this type of cartilage joint replacement or material study will better results with young patients as against older patients. Load distribution of the stifle, affordability and ease of animal handling, which type of animals we want to. If I am going to try it out on horse, it may be very difficult to do that sort of studies. There is a lot of issues involved because of the size of the animal. Rodent models are easy to use. The information about the residence time of an implant and also determine the type of repair tissue formed, what are the tissues that are formed. So, it gives you some knowledge. So, you may try to do a rodent model initially. Then you may go into rabbit model looking at cartilage repair, as they have larger joints when compared to rodents and are a good size for easy surgical procedures, specimen handling. So, we can look at cartilages of this thickness 0.25 to 0.75. If you go to large animals, okay, like dogs, pigs, sheep, you can do short studies, long studies, 8 to 12 weeks. This provides information regarding the biocompatibility, cellular response, persistence and condition of the implant within the defect. Longer studies, we can gain confidence in the extent of success in the repair, regeneration of the articular cartilage, including interface with adjacent cartilage and subchondral bone as well as the opposing articular surface. This is the ASTM standard on which these cartilage repairs are carried out using by different types of biomaterials. So, if you are talking about large animals, dogs, pigs, sheep, goats and horses are used. So, one can look at short duration, one can look at longer duration studies. Okay. So, what are the advantages, disadvantages of various in vivo models commonly used in the assessment of biomaterial strategies for cartilage defect? This is obtained from this particular reference. Okay. Mouse, low cost, manageable, easily available, transgenic available, that means we can create different transgenic species of mouse if you want to study. Can be used in subcutaneous and intramuscular, but then the joints are very small, so in situ examination is impossible. Rats, low cost, easily available, we can maintain in house, permanently open growth plates, accelerating intrinsic healing, increased density of cells in cartilage causing more efficient healing. But if you want to do partial thickness defects, then they are not good. Rabbit, low cost, maintain in house, increased intrinsic healing due to increased cell density, very different load characteristic, consistent partial thickness defects, but very difficult to achieve. So, you see good advantages, but disadvantage, good advantages, disadvantage, good advantage, disadvantage. So, if you are talking about small animal models for looking at cartilage, then you are having certain advantages and disadvantages, and selecting the correct animal is a big challenge. Again, if you are looking at large ones like dogs and pigs, naturally occurring disease state, because dogs also get this type of tendon issues, cartilage issues just like human beings. So, they do cooperate, thin cartilage, small critical size defect. So, we can study very small, but then the ethical issues are very, very problematic. Pig, biochemistry similar to humans, bone apposition rate and trabecular thickness similar to human. So, we can study very nice partial thickness defects. It is expensive, difficult to obtain at skeletal maturity, specialized habitat, they need very special habitat, they are very also very temperament. Okay. Look at goat, anatomy and biomechanics similar to humans, partial thickness defects possible, easily available, maintenance is also very low. Sheep, again maintenance low, anatomy similar to humans, partial thickness, defects possible, easily available. Look at horse. So, large defects similar to humans, partial thickness, large diameter defects possible, naturally occurring defects, similar biomechanics in trochlear groove, second look arthroscopy possible, but it is very expensive to acquire and maintain. We need very specialized centers, cannot avoid weight bearing on the joint during rehabilitation phase, very dense subchondral bone, MRI, CT were all very difficult because they are huge. So, you see small animals, large animals, many of them have advantages, disadvantages. So, there is a big challenge, especially when they do this type of preclinical studies before they really venture into human volunteers. Okay. So, next one is implanted associated infection. As I mentioned long time back, biofilm formation on implants and implant associated infection is a very serious problem as soon as a biomaterial is placed inside the body. So, you need to study these again in small animals. So, one can create this type of infection on a certain implant, surface biofilms, surface infections, and place it inside the animal models and see how the animal models develop. Okay. These studies are extremely important, and because as I said, implant associated infections are a serious problem on the first 1 or 2 days, and most of the biomaterials, 60–70% of the biomaterials fail because of implant associated infection. So, mice, rats, rabbits, guinea pigs are very good models to study that. So, advantages, less variability in standardized infection. So, we can create the same infection again and again and test it out on many of these animals. Many experimental models can imitate human disease. So, we can create, there are genetic models of mice which can be created with certain type of defect or missing gene to simulate human volunteers. Okay. So, genetic manipulation is possible. Many studies in human cannot be done because of ethical issues, whereas they can be done on say mice, rats and rabbits actually. They are also of course less expensive. Less time is required because of short lifespan and fast reproduction. So, if I am looking at what is the effect of infected biomaterial on their reproductivity or their future developmental stages, we can study easily on these type of animals because they have short lifespan and fast reproduction. What are the disadvantages? Matching pharmacokinetic profile may be difficult. So, if I am looking at exact elimination rates, exact degradation rate of biomaterial, it may be very difficult to match with the human volunteers. Matching side effects is difficult. So, the side effects these animals may get and the side effects the human volunteers who may get may not exactly match. Okay. So, that is a very serious issue. These are called disadvantages on animal studies. Okay. So, we will continue more on these animal studies, use of animals in biomaterial research in the next class as well. Thank you very much for your time.